Hello students, welcome to EPG Pathshala. I am Dr. Vibhadhavan from TERI. Today we are going to talk on module Somatic Hybridization under the paper Plant Biotechnology and Crop Improvement. Now in this paper we are going to learn about protoplast. Protoplast, which are naked cells without cell wall. How do we isolate protoplast? And protoplast, then thereafter, how do we fuse them? They of course have natural tendency, but we want certain combinations to be formed, so different technologies have been developed. And once a somatic hybrid or cybrid has been produced, how do you identify them? What are the hybrids? Are they cybrids? Are they homokaryons, heterokaryons, and so on? And thereafter, what is its application in crop improvement? Now, protoplasts are single cells with their cell walls removed, thus exposing plasma lemma or the cell membrane. Plant cells, they have a very rigid cell wall, while animal cells, they lack cell walls. So therefore, protoplasts are restricted to plant kingdom. Now, what kind of opportunities it gives you? Now, it's a cell without any cell wall, which provides strength, which also in a way restricts inflow and outflow of material from the cytoplasm. But here there is an opportunity that once the cell wall is removed, then there will be free flow of material from the cytoplasm as well. Now let me share with you that the possibility of forming cells without cell walls was visualized way back in 1892 by Clerker, who demonstrated isolation of protoplast by incubating cells in plasmolysing solution and manually cutting them with a sharp knife. But of course, this method was possible only for vacuolated cells, and the success rate was very poor. And in a way, no practical application was visualized at that stage, so it just remained one of the experiments. Thereafter, it was Cocking in 1960 who developed method for enzymatic isolation of protoplast. In the following years, in 1971, there was a report of complete plantlet regeneration from protoplast. And thereafter, in 72, it was that hybrid plant can be produced through protoplast fusion. So you fuse somatic cells with naked somatic cells and fuse them. That is what is somatic hybridization, and you can get a complete plant out of it. Of course, there again it was at the mercy what is happening. So cells will fuse themselves, but thereafter in 74, use of PEG or polyethylene glycol for protoplast fusion was successfully used, and again plantlet production in 1978, which was of intergeneric somatic hybrids. Now just visualize, like I have said, that protoplasts, they have tendency to fuse, and it is irrespective of the species. Now what happens in case of usual fusion or crossbreeding is that it is restricted. There are pre-post fertilization barrier. But here the potential is that it is a naked cell and another cell, so it won't realize what plant species it is. It can fuse. And when we were students, in a way it was taught to us, or rather that imagine a plant which gives you potato in the root zone and it gives you tomato at top. So that was the kind of vision that earlier scientists had. Of course, there were a lot of barriers thereafter and it never became that successful, but still it's a very successful technology. Now, use of chemicals like PEG, again, this does not identify, it only promotes protoplast fusion, but what we are also looking at, that can we develop a method where you plan giving one protoplast after the other and two different types, so that you are making hybrids of the desired kind. And that was achieved through use of electrofusion method. Of course, once you have developed these hybrids, the next possibilities are how do you evaluate them, how do you ensure that these things are or you are evaluating those hybrids and then taking further. And that is, fluorescent activated cell sorting method was developed between 1979 and 1984. Thereafter, computer directed in 1987, and in 1990, the first commercial cultivar of protoplast fusion was released. And in 1995, it's a very interesting research where Nicotiana protoplast was fused with a mouse protoplast, so plant fusing with the animal cell. And it has introduced animal traits in the plant, that is mouse immunoglobulin G in the leaves of Nicotiana. For protoplast isolation, the success came by the invent of enzymes, that is cellulase and meserozyme. And fortunately, it was not just the discovery of these enzymes and use in the crude form. Once they were identified, there were commercial companies who came forward for the commercial production of it. Cellulase and meserozyme, they have their origin from fungus and bacteria respectively, and they can be commercially synthesized. Now, initially, it was a two-step process whereby the cellulase, which is first used so as to degrade the cell walls and release protoplast, and meserozymes, which breaks tissues into single cells. So first you use meserozyme and then you use cellulase. But thereafter, the methods were developed where both of them were simultaneously used. So this shows you what are the different steps in isolation of protoplast. You take a healthy plant, you take the leaf that gives you good protoplast and cut it into very thin slices with the help of a razor blade. Thereafter, you transfer these slices into liquid containing the filtered enzyme solution or the commercially available enzyme solution. So then you incubate them for few hours, and then the cut leaves, they give you protoplast, which can then be isolated through the help of cut tips and transferred to whatever your next steps are, whether you want to fuse them or you want to grow them into plantlets. So this is the technique for protoplast isolation. Now, protoplast isolation is dependent on the explant. Usually we take leaves from in vitro form shoots or leaves of fresh growth. So you want healthy leaves, and in vitro, when we say, it is largely because they have more vacuoles, so it becomes simpler. For most systems, pH in the range of 4.7 to 6 is good. Of course, these enzymes are active between 40 to 50 degrees centigrade, but at these temperature, the explant cannot survive, or you get very poor yield of protoplast at those temperatures. So therefore, one is compromising by choosing a temperature of 25 to 30 degrees centigrade. Now, what happens is that when the cell loses its wall, it becomes very sensitive to in which media it is. So osmotic pressure, osmoticum of the media, that plays an extremely important role, because if the osmotic pressure, depending on the osmotic pressure, protoplast might burst or protoplast might shrink to a level, it loses all its water, that it cannot carry any further metabolic activities. And this osmoticum usually is maintained by sugar in the range of 400 to 800 micromoles. Protoplast, they have tendency to aggregate and fuse with each other. So even if you don't do anything and you isolate protoplasts and leave them, there is tendency that they will form colonies, they will fuse with each other. There can be 2, there can be 3 protoplasts fusing together. But then that is a way of producing protoplast, because it will be the same type fusing, it will be aggregates and so on. But you want protoplast to fuse in a planned manner. And for that, what we do is we increase concentration of ammonium nitrate, polyethylene glycol or PEG, that has been used extensively. And one of the best ways is to induce electric stimulus, and you bring protoplast of two different sources, one from each side, so that you get the hybrid. So it is surely a hybrid in which protoplasts of two parents are participating. Can also bind calcium in the fusion mixture, which may form a bridge between the PEG chain and negatively polarized group of the membrane constituents and thus favor agglutination. Agglutination of two or more protoplasts along large surface area results in tight adhesion of the membranes and formation of cytoplasmic channels where they are adhered closely. The channels gradually expand and the fusion body's cytoplasm starts flowing from one protoplast to the other, resulting in the mixing of cytoplasm of the two cells. The fused protoplast with nucleus of two different parents is called heterokaryon, and those with nuclei of the same parent are called homokaryon. The fusion of nuclei in heterokaryon during incubation results in the formation of hybrid cells. The frequency of PEG-induced fusion is influenced by the physiological stage of the protoplast, density of protoplast, molecular weight of PEG, calcium ion concentration, pH, osmolarity, and method of application of PEG. In the past couple of decades, electrofusion has become an alternative method of somatic hybridization. The method is rapid, safe, as treatment is non-toxic, and more reliable as it allows fusion of defined protoplast pairs. The electrofusion apparatus can either be fabricated or brought off the shelf because large number of vendors are producing it now. The technique involves two steps. Step 1 is aligning the protoplast, and second step is fusing the protoplast. The method involves aligning the protoplast and applying one or more pulses of high voltage DC current at an interval of half to 2 seconds. It results in the breakdown of cell membrane of the protoplast at several places, and especially those which are in close contact, resulting in fusion of cell membranes. When the fusion mixture is kept undisturbed for 30 minutes or so, it attains normal spherical form, and you again get largely formation of heterokaryons. As I have said earlier, this is a favored method over chemical method because of being safe, non-toxic, and high frequency. Now the figure on the right that shows you that you get different kinds of cells. You are bringing two cells of two cells from different parents, protoplast from two cells. They fuse. There is nuclei of the two as well as the cytoplasm of the two. Now the possibility is that these two fuse and you get hybrid formation. But there is also a possibility that nuclear material of one of the parents get knocked off and you only get the nucleus of one but the cytoplasm of both. And those combinations are called hybrids. It's only the cytoplasm which has fused and not the nuclei. The protoplast culture techniques are similar to the other cell culture techniques. So the very first factor becomes, after explant and the method of separation of cell walls, is how many protoplasts per unit volume are kept. What is the protoplast density? Now protoplast, once it's in cultures, it releases substances which promote growth as well as those which inhibit growth. So you have to draw the delicate balance where you're getting good fusion as well as good growth thereafter. We use hemocytometer to measure the protoplast density so that you can repeat your experiment. It's not hit and trial, but you work out for which species how many protoplasts should be there per unit of volume. Cultivation of protoplast, we follow the same principles as cell culture. We use Agro's embedded culture techniques, which is similar to Bergman's cell culture technique. We use liquid media on shaker protoplast of many species fail to divide on semi-solid media. And then one important thing initially, especially in the initial stages of protoplast culture, is that you have to maintain the osmoticum of the media. We use double layer method. It's semi-solid media at the base and thin layer of the liquid media on top, as well as agarose droplet beads method, so as to get localized area colonies of high cell density. Initial stages is to maintain the osmotic of the media so that the cell doesn't lose any water. And initially, because of the lack of cell wall, you also have to incubate these cultures, keep protoplasts in dark. Nicotiana tabacum, it was the first plant species which was regenerated from the protoplast way back in 1971. And by the end of the last century, there are 365 species belonging to 161 genera and 46 families that have yielded viable protoplasts and plants thereafter. We have 135 hybrids, hybrids with 125 different parental combinations. This shows you that it is not restricted to one genera. There is large number of species from which we have been successful till date, and there will be many more where we don't have methods today, but with more research we can get protoplast as well as viable hybrids which can be produced. It's also one of the things which you have learned in the past is that there are few species which are recalcitrant. There are genes which are responsible for good regeneration. So one of the things that protoplast hybridization gives you is that you produce a hybrid of a species which has good regeneration potential with a species which is recalcitrant. And the hybrid, in all probability, will be able to grow. And if it's a very wide cross, there is possibility that chromosomes or the nucleus of one will get knocked down and it will be only the cytoplasm. But then it definitely depends whether it's a cytoplasmic trait or it's a nuclear trait in that particular species, which will dictate whether you are overcoming recalcitrance or not. As I have said earlier, you have to identify ultimately these hybrids or hybrids, homokaryon, heterokaryons that are formed through fusion of somatic cell protoplast. Whenever you are using any chemical, you get lots of plants, lots of hybrids which are just of the parental type, because it is rather there will also be a tendency to fuse among themselves. But in case of electric fusion, what we do is we bring two protoplasts of diverse sources from different sites and make them fuse. It's only diverse protoplasts which are coming. Now, depending on whether it's a hybrid in which you get a tetraploid formation, or it is that the chromosome of one gets knocked down, you either get a tetraploid hybrid or a hybrid. In case of chemical fusion method, if you look at this figure, you have two kinds of protoplasts. So I'm just showing one with red chromosomes and the other one with blue chromosomes. So you will get fusion of both reds, both blues, or you will have one red, one blue. So, and you will also have protoplasts which don't divide. So you have undivided protoplasts, you have homokaryons, that is the two similar kinds of protoplast fuse, and you have heterokaryons where you have both different kinds of protoplasts being fused. In addition, you will also have protoplasts where it's only that part of the cytoplasm has been exchanged. You will also have protoplasts where the chromosome or the nuclear fusion is not complete. So you'll also have variety of nuclear and cytoplasmic combinations in this mixture. Several strategies have been followed to select the hybrid cells, such as use of biochemical markers for proteins, other nutrients, and antibiotic or herbicide resistance. Metabolic complementation is often applied in asymmetric hybridization. In this case, the donor protoplast is irradiated so as their chromosome gets fragmented. The recipient protoplasts are treated with metabolic inhibitors. Thus, protoplasts of both the parents fail to divide on their own. The homokaryons are also unable to divide, and it's only the hybrid cells which are capable of dividing due to complementation. To confirm hybrid nature of the plants regenerated, morphological, cytological, and molecular methods are applied. The vegetative and floral characteristics is the simplest morphological approach. Cytological analysis in terms of morphology and number of chromosomes, and use of molecular techniques such as rapid RFLP, SSR, AFLP, 5S rDNA, spacer sequence. These are the technologies which are frequently used. So, protoplast fusion, it may result in symmetric hybrids where hybrid cells have nuclear genome of both the parents, asymmetrical in which the nuclear genome of one of the parents may get partially or completely partially eliminated, and third type is hybrids where the nuclear genome of one of the parents gets completely knocked off but cytoplasm of both the parents is present. So these are the three main kinds. Now, what are the main applications in crop improvement? Usually when you talk of plant breeding, it is that you have to have closely related parents. We always look for resistance in the same species, in the same genera, and we try to bring that by crossing over. Now, it's not always possible because there are pre and post fertilization barriers. Pre-fertilization, when the pollen grain of unrelated species fail to grow on the stigma of another species. So, fertilization can never happen. The other is that you overcome that, or it is able to grow, it reaches the embryo sac, it reaches the egg, but again it fails to fertilize. So that is the post-fertilization barrier. So these barriers are well documented, or rather they are the one which inhibit progress in plant breeding or bringing traits from unrelated genera. Now over here, as we have mentioned earlier, that protoplast, they don't have any liking or disliking. They will fuse. Of course, there are problems that maybe the chromosomes of one will get knocked off, but the cytoplasm of the two will fuse. So the possibilities are enormous. You can get various kinds of combinations. The competing technology is that of genetic engineering, and that is, in case of genetic engineering, you will learn that you isolate DNA fragment which codes for a particular trait. You can clone that trait, and then through various means such as agrobacterium mediated or shotgun, you bring it to the genome of the higher plant. And the beauty is that this becomes integral part of the higher plant, and it divides, and it also get transmitted to the next generation. So there also it is that gene of any origin, of the plant origin, of animal origin, bacterial origin, that can be transformed into a plant. But genetic engineering has its own limitations, both in terms of achieving, because you really don't know where the integration will occur, as well as there are regulations in terms of biosafety, food safety, and it is still not an acceptable technology, especially for food crops in many countries, including India. You must be hearing a lot of controversy that is going on for the release of GM mustard, GM eggplant in the past, but we are not sure when they will be actually released. But in case of somatic hybridization, there are no such questions asked, and the hybrids can actually be released into the environment. Through somatic hybridization, we have been successful in releasing commercially important products with various species as well as various traits. For example, in the hybrid of Brassica juncea with Brassica oleracea, it has resistance to termic mosaic virus, Helianthus anus with Helianthus maximiliani, white rot resistance, Nicotiana tabacum with tabacum resistance to antibiotics, two different types of tabacum, Oryza sativa, bacterial blight resistance, Solanum tuberosum, tuber soft rot and early blight resistance, Solanum tuberosum, again with cross with Penicitum, giving rise to late blight resistance, Solanum tuberosum with Solanum ricci, antibiotic resistance, and Solanum tuberosum with Solanum tarni, potato virus white and late blight resistance. So students, I hope you have enjoyed this talk and you have learnt about a very exciting technique of somatic hybridization. Somatic cells, where you can remove the cell wall, the naked cells with just cell membrane, they can combine with protoplast of another cell, another type, and thus create unique combinations of nuclear and cytoplasm. Genetic enhancement can be achieved by recombinant DNA technology, but it is far better if you do through somatic hybridization because it's a far safer technology. You are carrying combinations or you are trying to bring traits from a related species. Over here, if it's very wide cross, the DNA gets knocked off of its own. Unlike genetic engineering where you push that particular gene to get incorporated. So here it is, there is still some natural selection to decide what kind of combinations should exist. We are still facing genome incompatibility. As I said, that the DNA of one genera may get totally knocked off, but over there also we are getting exciting cytoplasmic combinations. And I'm sure over years with more research, one will be able to overcome this genetic incompatibility barrier as well. And last but not the least, that a lot of commercial varieties have already been released and there are many more which are in the pipeline. So thank you students and happy learning.